Slide 1. Metalloenzymes or metal-activated enzymes. Hello everyone, welcome to Lecture 6. Today, we'll be diving into the fascinating world of metalloenzymes and metal-activated enzymes. I'm Mohammed Shahid Nadeem, and I'm excited to guide you through this topic. Let's start with the basics. Metalloenzymes are enzymes that absolutely require one or more metal ions for their catalytic activity. On the other hand, some enzymes are merely activated by the presence of metal ions. These enzymes play vital roles in essential biological processes like respiration, DNA synthesis, photosynthesis, and detoxification, utilizing common metal ions such as iron, copper, zinc, and more. One major type of metalloenzyme is the redox metalloenzyme. These enzymes have metal ions as part of their active site and participate directly in electron transfer reactions, catalyzing oxidation-reduction reactions. A prime example is cytochrome C oxidase, a key player in the mitochondrial electron transport chain, which contains heme and copper centers. Now let's talk about non-redox metalloenzymes. These enzymes also contain metal ions in their active site, but they don't directly participate in electron transfer. Instead, the metal ions contribute to structural stability or act as Lewis acids or bases in enzyme catalysis, like carbonic anhydrase, which uses zinc ions to catalyze the hydration of CO2. Slide 5. Let's zoom in on carbonic anhydrase. This enzyme contains a crucial zinc ion at its active site, tetrahedrally coordinated by histidine residues and a water molecule. The zinc ion polarizes the water molecule, facilitating the formation of a hydroxide ion that attacks carbon dioxide, ultimately producing bicarbonate. Now, let's look at hexokinase, an enzyme assisted by magnesium ions. Magnesium isn't part of the active site, but it's essential for hexokinase activity. It shields the negative charges of ATP, making the gamma phosphorus atom more accessible for nucleophilic attack by glucose, enabling the phosphorylation of glucose. Moving on to iron-containing enzymes, we have two main types, heme-containing and non-heme enzymes. A classic example of a heme enzyme is catalase. Catalase is an enzyme that converts hydrogen peroxide to water and oxygen. Slide 9. Catalase's mechanism involves the iron atom in its heme group. The enzyme reacts with hydrogen peroxide, forming an intermediate compound. Then, a second hydrogen peroxide molecule acts as a reductant, regenerating the enzyme and producing water and oxygen. Catalase plays several crucial roles in the body. It breaks down hydrogen peroxide, protecting against damage to lipids, proteins, and DNA. It also supports immune function by preventing excessive buildup of hydrogen peroxide in immune cells. Catalase also maintains red blood cell integrity and aids in cellular detoxification in the liver and kidneys. Furthermore, it helps maintain mitochondrial health and protects skin cells from UV radiation. Finally, it may play a role in preventing neurodegenerative diseases. Another important iron enzyme is cytochrome P450 or CYP. These heme-containing isozymes are embedded in the endoplasmic reticulum of hepatocytes. They contribute to the metabolism of many drugs, steroids, and carcinogens, primarily through monooxygenous reactions. Cytochrome P450 enzymes are named based on their cellular location and spectrophotometric properties. Over 30 P450 isozymes have been identified, with about 90% of human drug oxidation attributed to just 6 of them. These enzymes absorb light at a wavelength near 450 nanometers, when reduced heme iron combines with carbon monoxide. P450 enzymes have a wide range of functions. They are crucial in drug metabolism, detoxification, and hormone synthesis. They also play a role in cholesterol synthesis and fatty acid metabolism. P450 enzymes are also involved in vitamin D activation and bile acid synthesis. They metabolize steroids and modulate the body's response to toxins. Finally, they can contribute to cancer prevention by metabolizing potential carcinogens. Let's shift our focus to copper-dependent enzymes, specifically tyrosinase. Tyrosinase is crucial for melanin biosynthesis in animals and the browning of fruits and vegetables in plants. It's classified as a type 3 copper enzyme, relying on 2 copper ions at its active site. Tyrosinase can act on both monophenols and diphenols. It catalyzes the hydroxylation of monophenols to O-diphenols and then oxidizes these to O-quinones. These O-quinones are unstable and polymerize to form melanin in animals or cause browning in plants. Slide 19. Tyrosinase catalyzes the O-hydroxylation of monophenols, as well as the subsequent 2-electron oxidation of the resulting O-diphenols to the respective O-quinones, which non-enzymatically form melanins. Thank you for your attention. I hope you found this lecture informative and engaging. If you have any questions, feel free to ask.